Well, our next speaker is Boris Bernhardt. Boris is an associate professor of neurology, neurosurgery, and psychiatry at McGill University. He has a very strong background in image processing, in epilepsy, brain modeling, and particularly of late, gradient mapping, and characterizing the brain through gradient perspectives, both in evolution and in disease. So it's a delight. He's also a Canada Research Chair, I should mention. So I'm going to hand it over to Boris to tell us about dimensional perspectives on multiscale cortical organization.
Thank you so much, Alan, and the organizers for having me here. It's really a pleasure and an honor to speak. And I've just arrived yesterday, but today the talk that we've just heard from Dr. Corbetta was already very inspiring. So I hope to also tell you something interesting about the line of work that we've been doing in the lab. And as usual in neuroscience or in science in general, this is teamwork. So most of the work that I'm going to present is really orchestrated by a group of talented and motivated trainees. You see a few of them here: Jordan, who you'll hear later, Young-un, and Yuju, they're all here. And I've also benefited in my career from a large network of international collaborators, both conceptually and methodologically. I just want to highlight a few individuals who've been really key to some of the approaches that I'm going to talk today. Some of you may Daniel Margulies, Casey Pacola, from a postdoc in my lab, now in Jülich, as well as Rob Leach, Mike Milham, Sophie Falk, and last but not least, Johnny Smallwood, who has really been a psychologist working on the resting brain for most of his careers, and now really started to develop a conceptual framework in the neuroscience to study spontaneous brain activity as well. And we've heard already very interesting work from Dr. Corbetta about this before. Obviously, I'm also benefiting tremendously from being involved in HIBALL and from all the 3D models and resources that have been aggregated, including BigBrain. And we are also trying to contribute to these efforts by developing tools, but also by disseminating data sets. And you'll hear more about this at the end of my talk. So my talk will have 4 different sections. So first, I'm going to tell you a little bit about macro-scale cortical organization, how we can look at the cortex from a dimensional perspective. Then I'm going to focus on the hippocampus, which for some of you might be the favorite brain region, because this is really one of the model systems in the brain to understand the covariation of structure and function. And it's also a microcosm to understand principles of brain evolution, brain development, plasticity. Finally, I'm going to talk about the DMN, default mode network, a little bit of how we can use dimensional perspectives to understand spatial patterning of this really fascinating system, to potentially go a little bit more into what is the hardware of the DMN doing and how does this contribute to the diverse cognitive states that it produces. And then at the end, I'm going to give you something useful on the way, talking a little bit about the data and the tools that we've developed. So let's get started. So one of the key assumptions of neuroscience is that brain structure and function co-vary, and there's a long history of different studies that have tried to chart the cortical territory with respect to different microstructural features, including histology, myeloarchitecture, and so on and so forth. And overall, there's a strong relationship between microstructural properties of the cortex as well as functional specialization of different cortical regions. I work at the Montreal Neurological Institute, and some of you may know that Walter Penfield has really started a lot of cortical stimulation and recording studies where different functional specializations, for example, the homunculus and these things were discovered. There's a lot of body from lesion studies where different brain regions may be affected by pathology, and that leads to deficits in function. And then another line of work with which we can interrogate cortical function is through intracranial cortical electrodes in patient populations, for example, in patients with epilepsy, where we can start to understand different dynamic properties of different cortical areas using invasive recordings in patients. This obviously is something that we cannot do necessarily in healthy humans, or we shouldn't do it, so we have to resort to non-invasive techniques such as magnetic resonance imaging or MRI, which is basically my bread and butter technique. MRI is a very versatile machine, and it's very fascinating because it's a physical recording device of the human brain, and it gives you the ability to interrogate different aspects of brain structure, brain microstructure, as well as brain function in the same session. So you can do coupled structure function studies in the same individual, which is something that in the olden days of neuroanatomy was not per se possible because it's so laborious to do histopathological studies together with obviously antemortem recording investigations. So without going into details, there's several contrasts that are, for example, myelin sensitive from the Human Connectome project. You've heard, for example, about T1 over T2 ratio mapping. There's quantitative T1 relaxometry, magnetic transfer imaging. So all these contrasts are not one-to-one related to myelination, but they are sensitive, which means they correlate to some degree to myelin cholesterol in the cortical sheath. We can also, and Dr. Corbetta has talked about this at length, so I don't need to repeat this, but also interrogate the brain during different functional contexts, for example, during tasks, movie watching, but also in resting conditions, and then look at hemodynamic fluctuations of different brain regions, and then look at the coordination or the statistical dependencies between these hemodynamic fluctuations between different brain areas to map connectivity of specific regions. So here, for example, of the precuneus PCC, but we can also do decompositions of whole brain, large-scale network organization in order to derive what we refer to as networks of the brain. And this here is a canonical parcellation of the brain into different functional, intrinsic functional networks done by Thomas Yu and Fenner Kreenan. The human brain is very complex and it's very high dimensional, and when we do histopathological studies we can just appreciate and get overwhelmed by the dimensionality sometimes, and the same holds true for neuroimaging, where data representations become increasingly multidimensional, specifically when we start using multimodal imaging where we don't just have one contrast at hand but multiple contrasts. So there's different ways to deal with dimensionality, and just to give you a data science example, we can either cluster the data, so partition data into different subgroups, or we can look at eigenvector decompositions where we try to identify motifs or salient dimensions that explain the variance in the data set. The research program of cortical arealization is very long, and it's one of the key drivers of modern anatomical studies and also of computational neuroimaging, where we can extrapolate from approaches that have been pioneered by the Jülich group, for example, on histopathological data and translate them to in vivo human imaging. On the bottom, you can see, for example, the Glasser parcellation that was also mentioned earlier in the talk, where different in vivo contrasts have been used and where boundaries between different areas have been drawn based on local derivatives of feature change with respect to myelin changes, for example, task fMRI boundaries, as well as resting state connectivity. And here you can see the network that we've also seen earlier, where we can segregate the human cortex into these different systems, externally oriented, internally oriented, and so on and so forth. In my talk today, I want to talk a little bit about a complementary approach, namely these eigenvector-derived eigenvector decomposition, or also referred to as gradient mapping techniques. You can see Daniel with a chicken here, who has done a very high-profile paper in 2016 that has decomposed Human Connectome project resting state fMRI data into a set of salient dimensions explaining shifts or changes in local connectivity patterns as we traverse across the cortical mantle. And when we do these eigenvector decompositions, we can always classify the different components that we get in terms of explained variance. So we can talk about a principal component as the component that explained the most variance in the human functional connectome, which you can see here on the top. And what is really striking about this first principal component is it really shows this characteristic sensory to association or sensory to transmodal pattern running from the primary systems towards the default mode and the paralimbic systems. What is very interesting about these decomposition techniques is that they recapitulate a lot of the research that has been done by decades of invasive track tracing studies. So here you can see a schema that recapitulates the Marcel-Mesulam scheme of sensation to cognition that has been initially developed based on non-human primate work, but now been extended to human based on resting state fMRI by the work from Margulis et al. Another thing that is important when talking about these dimensions of cortical organization is that we can not only look at a principal component, but obviously also at secondary components. So other orthogonal axes that explain variance in the large-scale connectome, for example, a second gradient that differentiates more between visual and somatosensory systems, as well as a third gradient that differentiates the multiple demand and attention networks from other systems in the brain. So with this, we have a set of canonical axes with which we can try to explain the cortical organization. And what is really important here is that this data comes from connectomes, so 2D descriptions of interregional associations, and we compress them to a series of cortical maps. And this obviously makes things quite easy to conceptualize and also to situate different cortical areas in the cortical tapestry. Using these map descriptions, we can, for example, interrogate then these functional axes with other features, for example, brain structural features or features relevant to the geometry of the brain. And what is really interesting from this perspective is that it shows that really systems like the default mode network, they are topographically furthest away from systems interacting with the outside world. So giving a topographic basis for internally oriented or internally generated cognition, as well as decoupled thinking that, for example, happens when we are at rest and when we try to envision the future or think about things that are not related to our immediate environment, and that might ultimately also be human unique cognition. When talking about gradients, it's obviously also relevant what kind of gradients we are talking about. So gradients have been derived from parcellated data, but also non-parcellated data. So the technique is agnostic a little bit to the input feature. We also have to think about that we talk about scale, so we can look at gradients within specific structures. I'm going to give you an example about this when talking about the hippocampus. And we can use these macro-level gradient descriptions that I've just talked about before. We can also derive gradients from different features. So you've seen the resting state fMRI decomposition before, but we can also derive them from interregional measures of myelin similarity, for example, or from interregional measures of histopathological similarity and structural connectivity. And obviously the premise of these gradients is to ultimately explain function and cognition. So when combining gradients, for example, in the primary visual cortex, we can derive features that come from intrinsic functional connectivity, but that then resemble the topographic organization of the visual cortex. For example, eccentricity and polar angle of the visual field. Yizhu Wang, who's also in the audience and who will be very happy to talk about his work, has recently aggregated different functional, microstructural, and structural connectivity-based gradients in order to better understand large-scale cortical arealization approaches. So I'm not going to go into detail here, but you can talk to Yizhu, who's sitting right there. All this in mind, we can then use this dimensional axis of cortical organization in order to understand maybe a little bit where regions are in the brain and what is the relevance of this for function. And we can also then use this framework as a simplified low-dimensional coordinate system to, for example, interrogate structure-function principles in the human brain. I'm going to talk about a large-scale structure-function principle a little bit later, but this is just foreshadowing it, is that we can, in essence, derive very converging axes of brain microstructural and intrinsic functional organization when applying gradient mapping techniques. This is not black magic, so everyone can do it. We've developed a toolbox for this, BrainSpace, which you can download from the internet. You can use MATLAB or Python, so pick your poison, folks. And then it's basically, we have a functional connectome at hand. We derive a similarity matrix of connections. We do a decomposition to derive these components. We can plot them on the brain. Then we can use these components, for example, to sort connectivity profiles. Here you can see a microstructurally sensitive connectome. Here you see a functional connectome. It's documented and it's all available on GitHub. So if you want to use gradients, it's just 3 lines of code away. So it's really easy to use and it should be relatively straightforward. In the first set of studies that I'm going to show you, this is work done by Casey Pacola a few years back, former postdoc in the lab. What we did in essence is we derived these gradients from BigBrain. So this was our first encounter with BigBrain, and we are very grateful for Alan and Katrin and their teams to have made this fantastic resource available. I think we're all here also because of BigBrain. So we basically derived gradients from BigBrain. We derived gradients from microstructurally sensitive neuroimaging, and we derived gradients from resting state fMRI. Voila, all the gradients look the same, and paper done. So this is basically what we did. Here you can see a fly-through of the BigBrain. We possibly have seen this already yesterday, but I wasn't here. So BigBrain looks like an MRI, but it's not. It's a histology, very high resolution. What we can do from BigBrain, on the other hand, is we can derive similar image processing techniques as we do for MRI to derive these surface models between the inner and outer cortical interface. What Casey had then done is that she sampled vertically or perpendicular to the cortical interface multiple surfaces, and then we can sample profiles, so we can get intracortical profiles. We can then cross-correlate these profiles to look at profile similarity, derive a gradient based on this, and in this case we obtained a gradient that really ran from paralimbic towards more sensory and motor systems, so really a sensory-fugal differentiation in terms of microstructural laminar differentiation. This was also confirmed with literature-based atlases of laminar differentiation and cytoarchitecture classes, where you can really see this correlation between the data-driven gradient and these more literature-based previous descriptions coming from anatomy. We then used myelin-sensitive MRI, here based on HCP, but we also validated this with other contrasts like MT, like quantitative QT1, got very consistent gradients, and then used the in vivo MRI gradient as a bridge to interrogate structure-function convergence. And we indeed obtained, both at the group and at the single subject level, relatively consistent spatial pattern when looking at the resting state fMRI connectome gradient and the myelin-sensitive microstructural gradient. So all this then obviously creates a good framework to not only look at structure-function correspondence, but also as principles of cortical organization as they are being preserved or innovated throughout our lineage, evolutionary speaking, but also to use these gradients potentially as a coordinate system to understand aging as well as development. And then we can also interrogate these gradients at macro scale to understand plasticity and disease. So this is just an overview slide. I'm not going to go into details here, but there's obviously a lot of work from many different groups, including ours, that have started to harness these approaches in order to understand evolutionary, developmental, and disease-related principles of brain organization. That's the interim summary. So we now have relatively consistent evidence that we can use dimensional perspectives to better understand large-scale organization in the brain. Arealization is increasingly complemented with gradients. I think it's important that despite the abundance of work focusing on a principal gradient, there's multiple gradients in the brain, and I think in the next 5 or 6 years, or 10 years maybe, we are really trying to be interested in the coordination of different gradients and their overlap in explaining large-scale brain functions. We can derive these gradients from different substrates, including microstructure, function, as well as connectivity. And overall, this then really gives us a good framework to look at blueprints of brain organization as we move through the lifespan, but also throughout evolution. So shifting gears now and going to the hippocampus. So here we have, again, a fly-through through the hippocampus based on BigBrain. Really complex, interesting, unique structure in the brain. It is a cortex. It's an archicortex, sometimes erroneously labeled as subcortical, but it is a cortex. And what is really interesting about the hippocampus, it exemplifies a little bit of how structure and function co-vary, as I mentioned before, but it's also a nexus of different really important large-scale networks in the brain, including the limbic systems. It's at the end of sensory streams, so we talked about the ventral stream before, but it's also considered to be one of the key nodes of the human default mode network. So all this to say is that the hippocampus is important. And I have a sidekick in working in disease, epilepsy, but also many folks working in aging. So the hippocampus is one of the first to fall. So it's like a highly plastic region, but it's also one that's susceptible across multiple clinical indications. So we have a vested interest in trying to develop better models of hippocampal organization in order to understand structure pathology, but also dysfunction in disease. Here's work where we basically applied resting state fMRI gradient mapping techniques. So here you can see a little automatically generated hippocampal surface model in the brain where we can then get the hemodynamic signals from. We can also get the signals from different cortical areas, look at the hippocampus to cortex connectome, and then decompose this using gradient mapping techniques such as the ones from brain space that I mentioned earlier. Map the first principal component of the hippocampus. Here, this is shown in the center. So we can appreciate these smooth transitions between the anterior regions in the hippocampus and the posterior regions. And these transitions, they were consistent across all the different subfields, even if we did the analysis independently for them. So for subiculum, for the corona ammonis fields, as well as for the dentate gyrus. What we then cross-referenced these data with is that we also looked at meta-analytic co-activation, here based on Neurosynth, which is an openly accessible platform for task-based fMRI studies. So we got a very similar gradient with high spatial correlation, and we could also cross-validate this with manual segmentations of the hippocampal segments, head, body, tail. So overall, we see a relatively consistent anterior to posterior organization of the human hippocampus. When we looked at the second gradient, interestingly, it did not follow this anterior to posterior pattern, but it followed more the hippocampal infolding. So medial to lateral or proximal distal, depending on which school you're coming from to study hippocampal anatomy. And what was interesting in this, at the level of this data set, so Human Connectome Project, that also gives you different contrasts, including this myelin-sensitive T1 over T2 mapping contrast, is that we could see a relatively good correlation across the different subfields with overall myelination of the subfield. So overall, recapitulating, at least from a dimensional perspective, the subfield to subfield differentiation that we can see at the level of microstructure. When looking at BigBrain, so here, this is the BigBrain hippocampus. We did a similar work, again, led by Casey Pacola, where we looked at histology profiles of the hippocampus, and we also decomposed these profiles using gradient mapping techniques. Here, we could really get good confirmation for this mesial to lateral or proximal distal axis at the level of histology, so really showing both evidence for subfield to subfield organization, but also for a really continuous shift along the entire axis of the hippocampus. And why this was quite interesting from our perspective is that we could then relate these spatial dimensions of hippocampal organization, so this more anterior to posterior gradient, but also this mesio to lateral gradient, to the connectivity profile that the hippocampus has with large-scale networks, in this case specifically the default mode network as well as the multiple demand system, so that in the structure itself, there seems to be a topographic representation of large-scale functional motifs in the brain that could also help to explain why the hippocampus is so important in orchestrating large-scale brain dynamics. This is work by Jake Vogel, a former PhD student with Alan, now faculty in Lund, as well as Alex NGO, who's a PhD student in my lab. These are just two very complementary types of approaches, and I think yesterday you've heard a lot about the Allen Brain Atlas and the Allen Brain initiatives. So here what we can just appreciate is that we can also use spatial transcriptomics to recover similar spatial axes, in this case both anterior to posterior gradients, but also subfield to subfield gradients with respect to hippocampal gene expression based on postmortem data. And I'm just going to foreshadow a talk that's going to come by Jordan a little bit later today, HippoMaps, which is something that we've recently put on bioRxiv and that's currently under review. So where we really try to aggregate different data of the hippocampus in a common reference frame. In this case, we used automatic segmentation and analytical unfolding of the hippocampus, so to represent it as a 2D sheet. And we obviously represented BigBrain, but also other data sets coming from HIBALL, including PLI transmittance, together with in vivo features, intracranial recordings, as well as resting state fMRI connectivity, to really look at the hippocampal coordinate system and to use it as a data warehouse where we can store different types of normative hippocampal features. And here you can see, for example, how we can then look at interfeature similarities. And when we, for example, have an fMRI study that reports hippocampal activation, we can place our fMRI activations into this coordinate system and trying to find similarities between what we observe in our experimental conditions and what we have aggregated in this normative data set. So again, openly accessible, it's in Python, and you can also talk to Jordan if you want to use it. This is conceptual work by Sarah Guénon, where I was also fortunate to work with, together with Katrin, where it was really proposed as where this 2D coordinate system, or the importance of it, was really emphasized both with respect to the anterior to posterior differentiation, but also with respect to the mediolateral differentiation in terms of functional organization, in terms of microstructural organization, but also in terms of susceptibility to disease. We are currently working towards a perspective where we can then also use these insights, this again led by Jordan, where we can use these insights to kind of see with the hippocampal as an example how this can potentially inform bio-inspired AI in the future, with the hippocampus having quite unique properties and being like an example of very efficient learning, namely one-shot learning, which is a little bit in contrast to some of the large neural network models that are coming more from the AI field that are very data hungry and very resource intensive. So we've seen this earlier today. The brain uses a lot of energy, but it's still, these are still small structures that don't use as much energy as a supercomputer farm, for example. So there's a disconnect between what is biologically implemented and what is implemented with at least current AI systems. So just here to give interim summary. So hippocampus model system to study covariation of structure and function. We now have relatively robust evidence for anterior to posterior as well as proximal distal organization of the hippocampus. I've shown you some work where we can kind of also look at the associations between these two different axes, namely large-scale functional connectivity embedding and the microstructure of the hippocampus, and this can ultimately help us to understand larger scale systems to which the hippocampus preferentially communicates, including sensory processing, memory, limbic-oriented systems, stress reactivity, but obviously also the default mode network, to which I'll come in a second. So I possibly don't have to talk to you about what the default mode network is. So here, this is a canonical description of where we can find it in the brain. So medial parietal, medial prefrontal regions, lateral frontotemporal regions, and MTG. What is really puzzling about it is everyone knows that it's very important, but a lot of different functions have been ascribed to the DMN. So there's not a clear one-to-one mapping with respect to different functions. Multiple candidates have obviously been proposed, and these are often quite human-specific, including autobiographical memory, envisioning the future, theory of mind, moral decision-making. So things that we tend to believe are unique to our species. I've talked to you earlier about how we can also start studying the hippocampus, not just in terms of cognitive ontologies, but also in terms of its topographic location in the brain, where it's a system that's quite far away from networks interacting with the outside world. So this gives us, in a way, a topographic basis for why the hippocampus might engage in self-generated memory-driven cognition, why we are also able to decouple from the here and now, sometimes when, for example, we do planning, mind wandering, but why also the default mode network might be so relevant for higher order sensory integration. So it's not necessarily only a system, at least that's believed, that's really task negative, and there's many examples of studies where the default mode is actually quite important for tasks and where it also communicates with task systems, but also where it integrates sensory information from large-scale cortical networks. And its topographic position may allow it to do so because the representations become increasingly low-dimensional and increasingly abstract. Casey, so this is work that's about to come out very soon, has started to not just look at what is the location of the default mode network in the brain, but what might be the underlying architecture within the systems. And again, using our favorite resource, BigBrain, what we did is we derived a canonical description of the default mode network, and then we cross-referenced it with different forms of cortical types. So here, these are cytoarchitecture-driven cortical types. And what we could observe is that the default mode network is not characterized by one specific microstructure feature, but it's rather characterized by multiple different cortical features, including a lot of the eulaminate classes with a very balanced composition. So there's multiple types in there, including paralimbic cortices, but also more sensory-oriented granular cortices, so the eulaminate cortices. They are represented in the DMN, and the DMN, compared to all the other networks, seems to have a more balanced composition of different cortical types. So this is quite interesting because it speaks to the heterogeneity of the default mode and how it might participate in different cognitive functions. Using BigBrain, then, we also derived this data-driven axis. So here we derived a gradient just within the DMN. And to our surprise, we did not necessarily find such a clear and straightforward pattern as we would expect maybe just from cytoarchitecture studies, but we rather found a complex tapestry of different motifs within the subregions of the DMN. So what's the first observation that we can make here is that the different subregions of the DMN, they don't seem to be clearly differentiated with respect to the position on this data-driven axis. On the other hand, what we can see is that in some regions, such as the mesotemporal lobe, we can see evidence for a more clear and gradual pattern. So this recapitulates a little bit what I've shown you before with the hippocampal data showing microstructural differentiation along the iso to allocortical axis. But we could also see like really complex patterns that are more indicative of interdigitation between different cognitive networks in the default mode network. And while we don't fully understand the reproducibility of this, because this is only coming from BigBrain, what we could clearly see is that there were correlations between the microstructurally, so the data-driven axis, and the connectivity that the default mode has to other regions of cortex, where we have some regions of the DMN that appear to be relatively isolated from cortical inputs, and other regions of the DMN that receive convergent inputs, and in this case, the convergent input is parametrically modulated by the cytoarchitecture class of the input system relative to this axis. We use precision neuroimaging data to replicate these findings. So here we use 7 Tesla MRI repeated in a group of 10 subjects, and we could basically derive similar data-driven cytoarchitectural axes with similar motifs here again, showing the mesotemporal lobe structures and these more frontal interdigitated patterns, and importantly again, a correlation between the input into the default mode system and the cytoarchitectural diversity within it. Just to wrap this up, so the DMN was traditionally discovered as regions that undergo deactivations during externally oriented tasks, but there's now a lot of work showing that it actually has a functional role. So it's not just a passive system, it's really something that has a role. And in Dr. Cobetta's talk, we saw really interesting how intrinsic activity might be very relevant to prepare us for forthcoming situations and contexts. In the DMN, we can nevertheless observe, when we use static network analysis, cohesive network connectivity. We can see a clear link to the mesotemporal system, which obviously implicates a role in self-generated cognition, but also higher level perceptual integration. We can derive topographically informed perspectives on the default mode function, and I've shared some work with you that also shows that the hardware of the DMN also seems to be quite unique, yet complex. The complexity is actually what makes it so interesting to study. And when we look at trying to see associations between cytoarchitectural and microstructural complexity of the DMN and its input patterns, we can see at least a biological basis for potentially functions of the DMN that relate to higher order sensory integration, but also decoupled thinking, self-generated thought. So just to wrap this up with my talk, gradient mapping is something that's useful, has become my line of work in a way. We can use it to look at macroscale scans of trends of cortical organization, but we can also look at sub-regional organizational principles and microstructural patterning within specific networks, such as the DMN. It's obviously a very useful technique because we derive simplified low-dimensional coordinate systems with which we can look at structure-function associations, where we can look at biological principles of evolution, and we can also look at things like brain development and the emergence of brain hierarchies. And I've just briefly alluded to this. Obviously, using dimensional decompositions of the brain gives us some insights into how the brain might be organized at macro scale, and this can then ultimately inform the development and validation of bio-inspired AI. And to continue gradient mapping approaches, or in general computational neuroanatomy, we rely on resources like HIBALL, but it will become increasingly important, at least from my view, to have data that is really done in the same human individuals, so advocating for more precision neuroimaging data sets where we have both really high resolution descriptions of brain microstructure coupled with function in the same individual, ideally across multiple task contexts. So this is my segue to a data set that we have recently released. It's currently under review, but you can already download it. So this is a PNI data set, so also disseminated under the umbrella of HIBALL, was led by Donna Gift-Caballo, and Johnny Smallwood was really instrumental in helping us to design the study. So these are 10 subjects that underwent repeated 7 Tesla imaging at our institute. It's around 4.5 hours of scanning per subject. So it's a little bit like Midnight Scan Club, but at 70, we have really good and versatile microstructural imaging. So here 500 micron MP2 range data, T2 star, MT data. We combine this with structure connectivity data, resting state fMRI, and data across a lot of different task contexts. And this is coupled with annotation of the different functional states. So hopefully this can be useful for some of you, and if you have some questions, please ask us. This data is available in bits, so raw, but you can also get fully pre-processed, multimodal, co-registered data on the same repo. So yeah, if ever you are interested, go on OSF, and the link is here. This kind of complements a data set that we've released a few years ago. Jessica Royer was the main author of this, where we have a similar data set released at 3T, but not with the same fidelity. So this is rather, I mean, it's still like very high resolution, it's very broad, and it's also pre-processed, but it doesn't harness the full power of MRI by being 3T. All these data have been processed by MICA pipe, so there's a pipeline coming from the lab. Raul Rodriguez Cruces, he's the architect here of this pipeline, where we can take bits data, process it individually, but also merge it into a common representation. So you can get resting state connectomes, structure connectomes, morphological features, the microstructural similarity matrices that I mentioned earlier, so you can also get this from MICA pipe. And I think at the end, what's quite interesting about it, or what's quite useful maybe about this tool, is that you can have surface level, high vertex level data, so really high resolution data, but you can also have data parcellated into different atlases available on different templates. And brain space I mentioned before. So with this, I would like to wrap up. Thank everyone for your attention, but also thank all the fine people working with me on this and our colleagues and funders.